and yours are both two fascinating topics that have you managed to watch? Because I feel we've had some of your colleagues here today.
Yes, I haven't seen too much of it, but you've definitely had some of our colleagues today, so hopefully we won't be
Pathology definitely came up because we had a group who were here talking about the brain bank, and we had from Newcastle, and I don't know why I can't remember her name.
Lauren.
We had Lauren from Newcastle, who definitely looked at this and went, Lauren would totally have matched into this pairing. Lauren, but I wanted a brain bank group, but you all kind of work with the brain bank as well. So let's do some proper introductions. Lucy, why don't you go first?
Hi everyone, I'm Lucy. I'm a postdoctoral researcher at the Dementia Research Centre at UCL. My primary role is working on the Frontotemporal Dementia Prevention Initiative, and I'm also the study manager lead for the genetic frontotemporal initiative. So they're kind of big cohort studies, observational studies following people with genetic forms of FTD over time. And my particular research interests are around social cognition and social interactions between individuals with particularly behavioural variant FTD, but also across the different types, as well as looking at new and novel digital markers to be able to monitor the disease over time.
I can't help but think as well that I might have miscategorized you in my head. I had you in my head as fundamental scientist, and then when I was preparing the questions at the last minute, I went, oh, doesn't look like she's in the lab at all.
No, not in the I personally am not in the lab, but I work with a lot of people who are, and a lot of the pharmaceutical companies who are developing new drugs for frontotemporal dementia. So I do kind of fit, but I don't do the biological stuff myself.
Well, maybe we'll sell this as you can provide both sides of the story, because Tamron, you're very much in the lab. Why don't you introduce yourself?
Yeah, I am. Well, not so much now, but yes, I do try and stay in the lab a few days a week. So I'm Professor Tammaryn Lashley, Professor of Neuroscience at UCL. I'm also the Director of Research at Queen Square Brain Bank, which is the brain bank based at UCL. It's not part of the Brains for Dementia network, but it's a separate brain bank, and I'm also Vice President of the British Neuropath Society. So I've been based at UCL for over 20 years, feeling really old compared to some of the panelists.
Oh, so you've obviously got very generous lighting, because you can't tell. This is mine. Have you noticed Zoom has this feature where you can tell it on where it does this blend thing?
Yeah. So most of my work, well, yeah, all of my sort of work and my research group work is focused on looking at the post-mortem brain tissue, so people that have donated their brains for research, and I've spent many an hour looking down the microscope at what has gone wrong in these brains to cause the clinical disease that we know as either Alzheimer's disease or frontotemporal dementias. So I started off looking at two quite rare dementias, familial British and familial Danish dementias, that are caused by two different amyloids to the amyloid that we find in Alzheimer's disease, but sort of gave us clues as to the mechanisms that we do find in Alzheimer's disease. And then just because of funding opportunities, I think one of my first project grants was funded by what was then Alzheimer's Research Trust, which is now, as we know, AR UK, who have funded both my junior and senior fellowships to broaden the research on the different types of frontotemporal dementias. So I've had lots of collaborative projects over the years looking at what I call the traditional pathologies, so the A beta plaques and the tau tangles, but it's actually looking at the brains kind of from a different angle to see what we're not seeing in these brains. So obviously all around the world, and we've heard from panelists the amount of people looking at the classical pathologies, but I started to notice that there were other proteins in neurons or brain cells that were still there in the people that have had these diseases, that were showing different characteristics. And these proteins are a class of proteins called RNA binding proteins that are involved in processing our genetic information in the brain cells themselves, and can alter how that genetic information is then turned into the proteins. So that's sort of the main sort of driving force of my own sort of research group now, is to look at these RNA binding proteins and how they are affecting the pathology in frontotemporal dementia itself. But I've got so many different collaborative projects. We use the brain tissues to look for different biomarkers in the different frontotemporal dementias. We use it for PET tracers, to look at where the PET tracers that they use in clinics bind to in the path tissue themselves. So yeah, we ship tissue all around the world for various different projects.
I had a song in my head then. I don't know where this came from. And again, it's probably because I've been doing this for 12 hours, but my milkshake brings all the boys to the yard came into my head then. I have no idea why, except for all those collaborations come off the back of, because you've got the good stuff, right? I mean, you've got this incredible resource that is so unique. And those cases you mentioned earlier about the Scandinavian one and the Welsh one, they're the famous ones, right? Aren't they the original? Are they famous?
No, British and Danish dementia.
Danish ones, yeah.
No, so they're not the original mutations that John Hardy.
No, they're different.
They're different. So they cause. So the term amyloid is used quite loosely. Amyloid is an actual term for the structure of a protein, not the underlying protein itself. So as we know, in Alzheimer's disease, the amyloid protein is A beta. In the British and Danish, it's ADAN and A-BRI that cause the amyloid that causes the disease. So it's a different type of protein, but causes a similar kind of disease to Alzheimer's disease.
How on earth do you find that? How do you end up with making that connection between that one in the UK and the one elsewhere? And I mean, is it potential then that there are more of these elsewhere in the world?
So Chinese dementia has just been identified. Again,
like the Chinese flu, that's not quite the same thing.
They're mutations all in the same protein. So it was I was just in the right place at the right time. So I started as a research technician at UCL as the first patient had passed away and the brain was harvested with British dementia. Then the mutation was identified. My secondary supervisor at New York University made an antibody that then I could characterize the pathology within the brain. So I was just in the right place at the right time.
Well, there's more to it than that, but I understand where you. So that's fascinating, and I guess having that flexibility. I mean, you must, having that support. We're going to give a plug for Alzheimer's Research UK, but having that kind of backing of the charity funding and being in the right place, you've had this opportunity to kind of follow your nose, which I guess sounds like not everybody necessarily gets the same chance to do. Is to go, ooh, that's a bit interesting. I'm going to go look at that.
Yeah,
and having that independence.
Yeah, totally. I mean, I feel so privileged in a way to have had the support from Alzheimer's Research UK and Alzheimer's Society for so many years, and I guess I do see it as a bit of luck and in the right place at the right time. You constantly reading, researching the literature, working with collaborators to see where the next sort of hot topic is and what the sort of next avenue of research is. And sort of learning from other diseases as well. So, I mean, when I first started, there was no link between MND and frontotemporal dementia. Obviously, now we know that there probably is a spectrum of diseases. So genes and proteins involved with MND, ALS, overlap with FTD, but that wasn't even a thing when I first started working on frontotemporal dementia. So, things evolve over the years.
And I've thought of a tenuous link now. Because one assumes, because Lucy, you coordinate the FTD Prevention Initiative, which has these genetic FTD cohort studies. And I'm assuming that ultimately many of those people might go on to be donors.
Yeah, exactly. So a lot of it is interesting that you've just brought up about the Chinese dementia. We've actually been doing a lot of outreach work to Asia to try and increase the amount of research into genetic forms of FTD over there. So over the last 2 years, we've really expanded the cohorts to include sites in Southeast Asia, South Korea, where else we've gone, India, Australia, New Zealand, and Japan, and there's a few others as well. So kind of reaching out to those areas, trying to increase research there so that they can get participants involved in these types of studies, so that Tamron can do her amazing work and tell us all about what's going on.
It's a big network, right? I mean, it's that. So,
yeah.
I'm fascinated. How do you go about that? So you suddenly go, oh, we want to do that. How do you go about setting up and doing this? Finding these new international collaborators?
Yeah, so Professor Jonathan Aurora, who Tamron works closely with, works with the equivalent study in Europe that's in America called All FTD. So there's a big observational genetic FTD study out there. And they got together a couple of conferences ago and decided that actually it'd be a really good idea to pool all our participants together. Genetic FTDs, particularly rare disease, particularly when you start breaking down into the individual genetic groups. There's 3 main types, which might have been covered today, so apologies if I'm recapping.
No, do you know what? We've talked about FTD, but we haven't actually gone back to basics and explained why FTD is different to other things. So add that in.
So
just add that in.
A third of FTD is caused by particular genetic mutations, and they're in genes called C9orf72, progranulin, and MAPT or tau. And when you get down to that kind of individual level on the genetics, the number of participants available is very limited when we think about how we're going to enroll for clinical trials. And so by pooling all our resources together through the studies across the world, we're able to have as many people as possible going into those clinical trials. So John and Adam got together and set up the FPI and have started building over the kind of the last 10 years or so. And over time, research has obviously grown within different areas, and it's just a case of trying to link in with people and get them on board to the bigger picture and see that we really have a problem when it comes to the number of available participants for large-scale trials.
So let's talk about that for a minute then. So obviously that's a challenge because there just aren't as many people with that, or it's not necessarily that they're identified. What's your understanding of what proportion of people with FTD in the UK are actually in your database?
That's a really great question. I'm not entirely sure what the percentage is in the population that are participating in research.
So how many people do we think there are with FTD?
Oh, I don't know. I can't remember.
Tamron, have you got a guess? I don't know. Is there any published stats on this? Do we know?
Yeah. Do you know, Tamron?
I can't remember off the top.
I didn't mean to put you on the spot because everybody just remembers the Alzheimer's number, right? Yeah. It was 850,000 for a long time, and now I think it's 900.
There is published stats for FTD, but I think the problem with FTD is because it affects a younger age group. It's often misdiagnosed for other things. Yeah. And sometimes because the disease duration is quite a lot shorter than Alzheimer's disease, we might be missing quite a lot of cases.
And this is where Aitana and Nick and Amanda were talking earlier about having a blood test that helps you to disregard other diseases, then allows you to find your FTD people and potentially bring... Yeah. Ooh, something just beeped like it was getting... Is that one of you on me?
No, I didn't hear it.
Sorry, it sounds like something, when something sounds like it's getting unplugged and plugged back in again. Sorry. So the FTD, the blood test. So I guess the challenge there is that traditional pathways, aren't they? So somebody has a memory problem, they go to their doctor, and then depending on their age and roughly what they look like, they might get sent to a neurologist. But more often than not, if they're a bit older, they're going to go to their psychiatrist and the memory clinic issue, and then they're fairly barned out. It's only if people present with slightly unusual that they might go to a neurologist. I'm assuming God, I'm going to cringe when I say this. Neurologists would be better at identifying the signs of FTD than a normal memory clinic. No, are we allowed to speculate?
FTD is not normally memory led. It's obviously
so the chance of somebody with FTD ending up in a memory clinic is very unlikely anyway. They're more likely to have gone to neurology,
particularly in the early stages. Definitely.
But what are they going to get told they've got? Are they going to get told you have FTD? What are the other things, the pseudo things they might get misdiagnosed with? Yes,
they because as Tamron said, it often occurs earlier in life. So people are roughly in their 50s and 60s. It often gets put down to things like midlife crisis and kind of marital problems, and actually it's being underpinned by this change in behaviour and personality. And if
so put
they
down
do
to
get
a psychiatric issue rather than a
so they quite often end up in psychiatric care. Often they might be perceived to have depression or severe anxiety, or they may even have psychosis, particularly hallucinations and delusions in some types of FTD. It's quite common. So yeah, it can take quite a long time for them to end up seeing a neurologist.
I see. So that's a problem. So having the blood test we talked about earlier would be particularly useful, because particularly if it was cheap and readily available, you could just give that to everybody just to at least disregard FTD. That would make such a big difference, wouldn't it?
It would be useful if it was just one pathology, but it's not. So FTD can be split into 3 major pathologies, which is then further split into...
So this is where you were going, the different types. So we got to the genetic one. I rudely interrupted you before you got to the rest.
Yeah.
Keep going.
So the pathology, you can have the tau pathology, TDP43 pathology, or FUS pathology. And from that, there's different subtypes. So there's 5 different TDP43 subtypes, all with different clinical symptoms, all with different pathologies. So if somebody walks into a clinic with FTD, we have no way of knowing at the moment which one of those pathologies. There's no good having a treatment for a tau pathology if you've got a FUS patient, for instance, because the pathology and the biological process is going to be different. So we need biomarkers for those specific frontotemporal dementias, which is the tricky part.
Yeah, and I mean, I don't want to put you on the spot because I know it's not necessarily your field, but how far are we along with some of those? Is that even closely on the horizon?
We did a large study, me and John Rohrer. We went back and re-diagnosed about 100 cases from Queen Square Brain Bank, where they have really good clinical data. And from those, I think it was actually 95 cases, we could see that those with a sort of a semantic dementia presentation were more likely to have a type C TDP43 pathology. But we just need more numbers, more people to donate brains that we can look at to then correlate with the clinical presentations to refine the diagnosis further. So I think we're getting there. It's then identifying those biomarkers that underpins that specific pathology.
So
that's kind of why the trials are starting from the genetic side of things, because that correlation to the underlying pathology is much clearer than it is in those cases where they don't have the genetic forms. So we can be pretty certain that certain pathologies underlie certain genetic forms of FTD, and that's why it's the trials are starting from that point of view.
Is that because you can't tell the other ones until you've actually got access to their post-mortem tissue?
Mm-hmm.
Yeah. And none of the other scanning techniques have gotten sophisticated to allow for that?
Not yet. We're hopeful that PET might come and save the day.
But we've heard about Meg earlier. I guess Meg's know any of that. It's a different thing altogether.
No. We're looking for PET to save the day. I think at the moment we're hoping there might be some blood-based biomarkers as well that might be able to help us. But
so in the meantime, you compensate for that by having bigger numbers. The more people you can get, you can, but there just aren't enough volume of cases in the UK, even if they were all coming forward. So you have to grow these cohorts. And I mean, one would imagine places like the kind of Europe's pretty well organized, I expect. So there's one in Europe and the US you mentioned.
Yeah, so we've got I help John Rohrer run the Genetic FTD Initiative, which consists of 35 sites now across Europe and Canada, that's working towards observational cohort study of genetic forms of FTD. We're about 1,500 participants that are involved in that study, which is incredible, really. And then similar sort of numbers in the US in their study, but other areas, not quite as many participants involved at the moment.
And obviously you can see how other forms of dementia will benefit. As it is, each form of dementia makes a progress in its own research area. Everybody benefits. But is there any, any more progress? Would you say when it comes to looking at Alzheimer's compared to FTD, are you behind Alzheimer's or are you ahead in some areas? Is that an awkward question? It's not a competition. But...
No, it's not a competition. I would say it is behind because we're still learning the underlying proteins that are involved in the diagnosis, whether those proteins are the causative factors of these diseases. As with Alzheimer's, obviously we know the recent trials that are seemingly successful that have been in the news recently this week. But I think whether A, beta and tau, obviously there are the genetic driving factors, but we also see the pathologies, A, beta and tau pathologies in an elderly population that don't have any clinical symptoms as well. So what is the tipping factor to cause the clinical symptoms with Alzheimer's disease? I mean, we have controls donated to the brain bank. I would say, I think we have over 150 normal controls, cognitively normal controls, and out of those, only 10 have no pathology whatsoever. So what has...
Wow, that seems like not very many.
What has sort of prevented those that have the tau and A beta from having the clinical symptoms of Alzheimer's disease? I think it is something that needs more investigation, but it's really hard to get funding to look at normal people with pathology without the clinical symptoms. Okay, the pathology isn't as severe as what we see in Alzheimer's disease, so whether they would have developed Alzheimer's disease if they'd lived longer, but some of these cases are in their 90s and 100s and haven't been referred to a neurologist, so clinically normal with some respect, as you can be at that age. But even if we could push back everybody 30-40 years, then that's going to help dramatically. But it's often sort of just ignored that this A beta and tau deposition happens in the population, elderly population as well, with no clinical symptoms.
And can you get do you have a study running like that right now? Is there like a brain donation for those kind of super ages, the kind of plus 95-year-olds, plus 90-year-olds, where you've got a study going that wants to collect brains from those people to look up the difference? More of those. Sort
of running a side project, because I can't get funding for it. We just fit it in well in and around other projects.
Come on, funding. I can't help but think that this is something that Race Against Dementia, that's a bit of a change.
There are, I think the Netherlands Brain Bank has they called it the oldest old cohort. So they're looking at those that are above 100, at what those protective factors are, that hasn't tipped them over the edge into Alzheimer's disease. So for me, you have the plaques, the tangles, you have the genetic predisposition to Alzheimer's disease, but what is that third factor that tips it over the edge into? Is it the neuroinflammation, which obviously we've heard about microglia and other people working on the microglia as well. But there definitely is, for me, looking at the human brains that have the disease or don't have the disease, there is some other things that we're not visibly seeing or haven't seen yet because we're not looking at the right things.
And I'm guessing you also then want to look at the people who, am I right in thinking that the people that have the genetic version don't all necessarily go on to develop FTD?
No, they do. So if they have the genetic mutation, they will go on to develop FTD in their lifetime. There is a number of cases where they perhaps pass away earlier than the onset of their disease. So it does kind of look like it's not full penetrance, but the likelihood is that everybody who has the genetic mutation will go on to develop the disease in their lifetime.
But not in Alzheimer's disease, so there are rare cases that don't.
No, the APOEs don't all go on to develop that. Fascinating. This is so interesting. We could have easily gone on for another half an hour, I feel sure. But I'm happy to keep going, but I feel guilty because I'm taking up your Friday night, so I'm not gonna keep you, but I will give you a chance. Is there anything? So I'm guessing the big thing for you, Lucy, is if anybody's watching now who knows somebody or has a relative or knows somebody with FTD, ask them if they're involved in research, and if they're not, then where do you want them to go?
Yeah, so those people who are...
There might not, I know it's very unlikely, but this will live on YouTube.
Who knows?
This recording will live on YouTube for a lifetime. And so anybody who's watching can still, this news will be useful, not just the 7 people that are still watching on Friday night.
Yeah, so for those people with outer genetic cause for FTD, there's a number of observational studies that we run at the Dementia Research Centre. So if you pop onto the Dementia Research Centre website, there's links on there to kind of self-refer to the research groups. If anybody has a genetic form of FTD and are interested in observational studies, then you can contact us at genfi@ucl.ac.uk. So that's GENFI, Genfi. And if you have a genetic form of FTD and you're interested in participating in clinical trials, you can contact the clinical trials email address on the Dementia Research Centre as well.
And what about if you're just could keep adding to the list of what you want people to do, but what about if you're worried about FTD? And what about if you're kind of somebody who's heard some information today, you've looked on the website, you're worried, I'm assuming you're going to encourage them to go to their doctors.
Yeah, straight to your GP. Any kind of changes in behaviour, in your language, some people with FTD have difficulties with their speech and their communication, and they can also have difficulties with their movements, as well as having some of these psychotic symptoms as well. So if these are occurring much later on in life, in kind of 50s and 60s, and there's no other kind of explanation for those things, then do go and speak to your GP if you're concerned.
So even if you haven't got the memory problem, if you have those symptoms, do go ahead. And do you know what? Sit there in front of the GP. Keep pushing. Say, would you mind having a look in for FTD? Because I've heard about it. And that's all too often is that's where until you raise it.
It's not that well known. We are trying our best to spread the word, but some GPs still aren't aware of it, or other healthcare providers aren't as familiar with it. So there is a lot of information on our lab's webpage, which is ftdtalk.org. You can find loads of information and fact sheets on there about the disease and the illness.
And they've been mentioned a couple of times today, but the rare dementia support group as well. I'm assuming that they're involved.
Yeah, lots of information on there about all different types of rare dementias, and they're a fantastic team, and would certainly be able to help and point people.
I think our next fundraising effort will be for FTD, for the rare dementias group. But
that would be wonderful.
Thank you, Lucy. And Tamron, do you want brains? I don't mean in the in the zombie kind of sense.
Yes, I would, but no, because what we need is people to go through the Dementia Research Centre, to have those clinical records that we can then correlate with what we see under the microscope. I wouldn't say a brain without the clinical history is useless to us, but we just don't have the resources to take every single brain that we would like to take.
Are there specific? Is your brain bank looking for specific people right now that would? So yeah, we
do later. Yeah, we do collect a specific cohort. So mostly the cohorts where people have been seen at Queen Square themselves, through other charities, the Progressive Supranuclear Palsy charity and other specific cohorts. But likewise, if people think they are involved in any of those, they can ring our administrator, Lynn Haddon, and she will direct them, either sign them up to our brain bank if they do belong to one of those cohorts, or direct them to another brain bank that would be able to take their brain should they want to donate their brain.
And if anybody is involved in any of our research studies at the moment and are interested in doing it, you can speak to the research teams as well, and they'll be able to pass on the details to Tamron and the team.
And as we've heard, not just from Tamron today, but from other researchers as well, the brain tissue you use is massively helpful. It can make a great difference. It's the only way we can really understand this until we can get better with the scans and the other things that come along. Thank you so much for making this tonight for our very last session. We've got Professor Tammaryn Lashley. and Dr. Lucy Chisman-Russell, both from University College London. Thank you very much.
And thank you, Adam. It's a fantastic day, and you've done a really great job, and you've made it to the end. So well done.
I'm going to cry. No, not really. Thank you very much. I'm gonna do my close down.
Yeah.
I have another camera set up here that I've not used all day with a different angle to make it interesting. It's gone off. I'll have to do it to this one. Thank you so much.
Thanks,
Adam.
Adam. Bye.
Bye, Lucy.